Today we're going to begin to learn about protein biosynthesis. We're going to start with a concept that we learned in Chapter 1 called flow of information. If you recall, genetic information is stored in DNA, and it must be removed and rewritten into a nucleic acid called RNA, ribonucleic acid, and then that information is translated into the protein. The process by which DNA transfers information to RNA is called transcription. The information, once it is put into the RNA, then is translated into the protein. RNA may be, if you recall, a messenger RNA, a transfer RNA, or ribosomal RNA. All of these may be transcribed. Only the messenger will be utilized to make the protein. The protein is synthesized on an organelle of the cell, which we call a ribosome. Please review the structure and functioning of ribosomes in Chapter 6, which is our cellular chapter. Figure 17.3 is our next slide, and this is an overview of the roles of transcription and translation and the flow of genetic information. Examine the cells above and notice the location of transcription and translation in each. Information flow in the bacterial cell, which is a prokaryotic cell, is not restricted by a nuclear envelope. Therefore, there can be simultaneous transcription and translation in the cytoplasm. In the eukaryotic cell, transcription occurs in the nucleus, while translation occurs in the cytoplasm. Also, in the eukaryotic cell, there is a pre-messenger RNA or a primary transcript that is generated or synthesized first. It must then be processed. This is called RNA processing, which is really an editing process whereby the messenger RNA that has been synthesized must actually have the introns removed and the exons connected together. In addition, a cap and a tail are put on the processed molecule, and then it is ready to leave the nucleus and go to the ribosome for translation. In the next slide, before we examine the details of transcription and translation, we should probably discuss the genetic code. The following are important points regarding the genetic code. Number one, the genetic code is a triplet code made up of codons. A codon is 3 RNA nucleotides that code for one amino acid. There are 3 stop codons in the genetic code and 1 start codon. In figure 17.5, you will see the codon table for messenger RNA. The genetic code contains uracil in some codons, and recall that uracil is used in RNA instead of thymine. The messenger RNA nucleotides are read in sequences of 3, which is a codon, and each codon stands for a specific amino acid. The amino acid names are abbreviated in this chart, and you may wish to return to Chapter 5 to see their full names as well as their structures.